The broadcast is now starting. All attendees are in listen-only mode.
Good day to everyone joining us, and welcome to today's X-Talks webinar. Today's talk is entitled Gene Therapy for Rare Diseases, Strategies to Drive Operations. My name is Ryan Muse, and I'll be your X-Talks host for today. Today's webinar will run for approximately 60 minutes, and this presentation includes a Q&A session with our speakers. Now, the webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit your questions and comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel, which is on the right-hand side of your screen. And if you require any assistance along the way, you can contact me at any time by sending a message using this same chat panel. At this time, know that all participants are in listen-only mode, and please note that the event will be recorded and made available for streaming on xtalks.com. At this point, I'd like to thank PRA Health Sciences, who developed the content for this presentation. PRA Health Sciences delivers innovative drug development solutions that improve patients' lives, including their proven commitment to developing the gene therapies for rare diseases patient communities. Their people are passionate about clinical research, working tirelessly to provide quality results and share their partners' mission to develop advanced therapeutics to address unmet needs. PRA offers exceptional experience across all phases and therapeutic areas and a broad spectrum of solutions ranging from full-service clinical development to a pioneering embedded model. With 16,000 plus employees covering 90 plus countries, they bolster an impressive global presence with keen local insights. Their project teams harness their understanding of local regulations, standards of care, and cultural customs to effectively align approaches with each study's unique goals. At PRA, they love what they do because they are making a difference in the lives of patients and their family members worldwide by driving development of meaningful treatments. Over the years, PRA have contributed to the development of numerous drugs now available to countless patients. From their scientific and medical experts to therapeutically aligned project managers and monitors, they provide the commitment and expertise needed for today's complex studies. Now I would like to introduce our speakers for today's event. Amy has a comprehensive understanding of drug discovery and drug development from the research laboratory to patient bedsides from 20 plus years of experience in academic institutions, pharmaceutical companies, and CROs. She earned her doctorate in molecular and cellular biology and received postdoctoral training at Memorial Sloan Kettering Cancer Center. She has contributed to the discovery and development of new treatments in a wide array of indications, from rare cancers to development of gene therapy for rare neurodegenerative disorders. She is passionate about enhancing the efficiency of clinical trials through cooperative productivity and patient-centered design. She has proven methods for bringing meaningful treatments to the patients who need them. Chris has advanced degrees in biology, public health, and biosecurity from the University of Missouri and St. Louis University. Chris is chairman and CEO of Sabai Global with subsidiaries of Clinical Biosafety Services, Castle Institutional Review Board, and Shield Consulting. He serves on multiple advisory boards to gene and cell therapy organizations, and Sabai Global supports over 200 plus sponsors on over 200 plus unique gene and cell therapy protocols in the U.S. and external of the U.S. For Christine Wagoner, founding and running Cure GM1 is a true labor of love to help bring treatment to all those affected by GM1 gangliosidosis. Rare diseases affect 1 in 10 people, and there is no other 501c3 entirely dedicated to GM1. Cure GM1 funds medical research such as AAV gene therapy, enzyme replacement therapy, chaperones, inhibitors, and substrate reduction therapies. Cure GM has raised over $3.3 million to date through social media and grassroots fundraising. And so now, without further ado, I would like to hand the mic over to our first speaker today. Amy Raymond, you may begin when you're ready.
Thank you so much, Ryan. Hello, everyone. Amy Raymond here from PRA Health Sciences. And on behalf of all of our panelists, I'd like to start by thanking you for making time to be with us today. As the three of us worked together to develop this content, our goal was to leverage our experiences for insights that could help drive upcoming rare disease gene therapy trials. We know from the attendee list that with us today are folks who come from drug development, the clinical research sites, as well as patients representing their communities. And our aim is to provide information that can be helpful to each of those perspectives. Next slide, please. And I suspect most people joining us today are well aware of the context, but for anyone newer to the space or to help set the stage, I just want to take a moment to note how critically important gene therapy treatments are to these patient communities. There are roughly 7,000 rare diseases and hundreds more being identified every year. Half of all rare disease patients are children, roughly a third of whom will not live to see their 5th birthday. Collectively, however, rare disease is not rare. There are an estimated 30 million rare disease patients in the US, with a comparable number in Europe. So that's 30 million families worried about and potentially caring for a rare disease patient, which is a staggering number. But no less important, though, the economic impact of having that many citizens unwell is massive. It's worth thinking about the experience we all had over this last year of hoping there would be a COVID treatment in the clinic if we needed it, but not having it in the early days, of needing a COVID vaccine, not having it for several months, a historically short period of time. Even today, maybe having approved vaccines that maybe not everyone can access yet. All of those being a small taste of what it's like to be any of the patients from these rare disease families, given that 90% of them have no approved treatments. There are many, many rare diseases where gene therapy could conceivably be a meaningful treatment. The geography of developing new treatments of any kind is inherently challenging in the rare disease space. Approaches that necessarily bring a patient to the same clinic over and over for a long period of time has never been a great fit for rare disease drug development, and is frankly part of why we only ever enroll a small percentage of each patient community, all of which leads to slower and more expensive development, in turn delaying getting that complete data package to the regulators, ultimately though delaying or even preventing some patient communities from having an approved medication in the clinic. No matter which indication or even which technology is being leveraged to develop a given gene therapy, whether it's an AAV vectored in vivo gene replacement or a lentivirus vectored ex vivo gene therapy, or any of the many gene editing technologies that we're seeing become more widely used to develop rare disease treatments, in all cases, the interventional dosing trials are sandwiched between getting and having a firm grasp on the natural history of the disease, hopefully just the once, and later getting that same firm grasp on the longer-term risks and benefits of receiving that gene therapy. As Christine and I worked together to develop this content for the discussion today, it's really this last point we focus on, the unique stakeholder dynamics of developing gene therapy for rare diseases. Much of what we'll cover today could actually be applied to any rare disease drug development program, no matter the modality. But the risks and benefits, that dynamic in gene therapy, is of course a little bit different for rare disease drug development. On the one hand, the family sees an awesome potential for durable treatment, but on the other hand, that same potential for longer-term risks, including not being able to take part in subsequent clinical trials. If you're part of a community where clinical research is your care option, that's a lot to weigh. Every gene therapy program is aiming to do what has typically not been done for each rare disease community, provide meaningful disease-modifying, potentially durable treatment, but getting there in each case means employing strategies that can be new for some of us, but in each case are about pre-aligning the missions of the key stakeholders at the table with the aim of turning what could be time-consuming friction into momentum instead. What we see on the next slide is the traditional drug development paradigm, right? Starting on the upper left, the sponsor has a complete protocol, it's approved by the regulators, then starts working with a CRO, usually, to then identify sites. They go through an approval process themselves, and then that site is responsible for recruiting participants to that study, knowing that there will be something of a burden of participating. Ultimately collecting data, which is reviewed and analyzed, ultimately provided to the regulators. So this is being primarily driven by the sponsor and regulators as stakeholders. What we saw during the pandemic, however, is that as we all struggled advancing clinical research programs during that pandemic, that when we needed to, we could retrofit even existing studies to some degree in order to collect clinical trial data somewhere other than in the research clinic. And we were doing whatever we could to make the study safe to participate in under those circumstances. And by retrofitting, some of the solutions were pretty clunky, but today we can now take those experiences and apply them planfully in upcoming gene therapy trials. So while it's appropriate that sponsors and regulators have been key stakeholders, that won't change. As we'll see on the next slide, they are not the only stakeholders. What we need to do is bring the other stakeholders to the table, patients and their care circle. The fine details of exactly which solutions are needed in each case for each gene therapy trial to make study more achievable will be unique as each trial. But the strategy of recognizing these stakeholders and gather input to de-risk the study will apply in each case. Certainly the travel expectation, the distance, and the frequency of a standard clinical trial is significant, and for many families, an insurmountable challenge. Anything we can do to reduce this travel burden is helpful for making study participation achievable for these rare disease patient families. Recognizing that patients and their families are a major stakeholder in our drug development means involving them in our design and aspects of operations that will impact them directly. So using digital health technology and mobile health platforms to collect data remotely without those participating families necessarily being in the research clinic to collect that data is achievable. All of those considerations are aspects of what we call decentralized clinical trials, collecting high quality, compliant data outside of the research clinic, which does not need to be an all or none proposition. The tools and techniques of decentralization are not brand new. Many of them are approaches we've been trying to advance for years. But the experience of working as we did as a global community to try to continue developing new treatments for all kinds of indications over the past 12 months has lowered some of those barriers and has given us a chance to think more creatively about ways we can stretch without sacrificing the scientific integrity of trials while also producing interpretable safety and efficacy data. Next slide, please. Here we have a poll question.
Yes, so we have a poll question for audience members to interact with in real time. This should be appearing on everyone's screen right now. You can click on any of the answers you see in front of you and then click submit. The question we have for you is, what is the likelihood of decentralized clinical trials increasing in the next 2 years? Your answer options are very unlikely, unlikely, neutral, likely, or certain. Again, the question is, what is the likelihood of decentralized clinical trials increasing in the next 2 years? We'll give you a few more seconds to consider your answer and provide one to us and participate. We thank you all for your participation, and it looks like most of you have participated, so let's take a look at the results. It looks like 49% have come in for likely, 20% for certain, 16% at neutral, 10% for unlikely, and 5% for very unlikely. Well, thank you very much for your participation. It's back to you, Amy.
Thanks so much, Ryan. There we go. Now I can see the slides again. So taken together, everybody in this crowd who just voted, roughly 70% of you think that it is likely or certain that we will see an increase in decentralized clinical trials, which fairly well mirrors what was seen in a large survey conducted last year by Informa Connect, where they were asking, what effect did COVID-19 have on adoption of decentralized clinical trials? Roughly 3/4 of them felt like it did have an increase. Looking forward into the near future, we can see that the most respondents, and I would say 85%, our crowd today is perhaps less optimistic than the global crowd. 85% of the industry respondents said that they think an increase in decentralized clinical trial adoption is likely or certain, which to me is very good news for gene therapy trials. But how do we start thinking about the design of decentralized clinical trials? As we'll see on the next slide, what we need to start with is the question of what are the needs of all the stakeholders at the table? Then we'll get to the second question in just a moment of how do we apply this specific approach to specific scenarios. Lowering the activation barrier for all of the stakeholders means pre-aligning the needs at the front end. And by aligning those needs of patient families with our operational plan and protocol details, we make study participation less burdensome, providing a positive impact on that risk-benefit ratio I mentioned in the beginning. Provided that we're looking to collect high-quality data in a compliant manner, what we've seen over the last year is regulators have proven themselves thoughtful and appropriately flexible in terms of collecting trial data safely through this public health emergency. And we continue to see guidances from regulators about clinical trial modernization. So today, what we need to be asking ourselves and each other when we're proposing to do a gene therapy clinical trial, are we proposing to do something that has been done before simply because it's the way it had been done before? Are we making the assumption that it is what the regulators will require rather than moving forward on that assumption? So as a side note, one aspect that is somewhat unique with respect to gene therapy for rare disease is the, honestly, complexity of access to a gene therapy if it should be approved following all of those clinical trials. And it's a concept sufficiently complex that it certainly merits its own conferences to discuss at length. But just to touch on it briefly here, that while it does require early planning, it's entirely possible to work with a team who can help map out that real-world evidence package that will be needed for those later payer conversations. And in some cases, even use a HIPAA-compliant, transparently consented mechanism for passively collecting information about the entire healthcare experience for study participants, what they're having outside of the clinical trial for a robust real-world evidence package that doesn't rely on burden from either the site or the participant. But getting back to that larger question of meeting the needs of all stakeholders, on the next slide we see that the customized solution for making any particular gene therapy trial achievable for the patients and their families, which is largely the degree to which we decentralize it, is a function of many factors. The treatment population, of course, being a key factor, what phase of development we're looking at, whether it's a dosing phase or a long-term follow-up, and every single case, every trial we design has to be done in compliance with the local laws and regulations. Getting into the nitty-gritty, the data journey, the data we collect and how it moves in that trial needs to be mapped to the patient journey, which is now a little bit different because it's decentralized. And what we have seen over the last year is a huge uptake in use of digital health technologies, home health care, and increasingly we're able to rely on the local healthcare system to leverage some of that data for clinical trials. When I talk about the treatment population, what do I mean? On the next slide, we look at, the treatment population is, of course, a major factor for the regulator perspective. And a key input to the trial design, but that design and how we operationalize that design in turn have a major impact on which members of a rare disease patient community can take part in that clinical trial, how many of those patients could take part, and how many will take part. And I'm specifically referring to disease progression. Is it a disease that moves very rapidly? What age of patients we plan to treat in the trial? The patients themselves are also thinking about whether or not this trial is their only care option. And digging into the finer details, it's also a function of what we are measuring in each trial and how we measure it. There are times when we absolutely need to be using a manual measurement in the clinic done by an expert clinical evaluator, but this should be supplemented, of course, with the patient experience data that regulators are asking us for, those patient-reported outcomes, the use of digital health technology, and for some indications, objective data, the lab results. With more than safety, it can talk about efficacy as well in some cases. And as we see on the next slide, where on each of these spectrums a particular trial falls doesn't necessarily dictate, but does have a major impact on the degree to which we can decentralize a clinical trial. none of us today are looking to fully decentralize a gene therapy trial, right? We may need to have the participant in the clinic to receive dosing, and we certainly want to ensure proper safety monitoring during the critical window following dosing, but we don't need to have every patient back to the clinic for every single data point. The more we can fit these trials into the complex lives these patients and their families are really living, the more achievable we make clinical trial participation. So, having laid out the framework, I want to skip pretty quickly over a couple of specific examples so that later you could consider how the framework applies to your own upcoming clinical trial. On the next slide, please. We'll look backwards to one of the first approved gene therapies for a rare disease, not the first, but one of the first. A gene therapy to treat spinal muscular atrophy. So if you are not familiar with it, SMA is a devastating disease where a child is born seemingly healthy, and the first signs of trouble come when they fail to meet these early developmental milestones. Being a rare disease, unless there's a family history, it can take some time to come to a proper diagnosis. Prior to gene therapy being developed and approved, the diagnosing physician had to deliver what was essentially a death sentence, right? Telling parents that their child would die by age 2. Point being, it's a classic example of high unmet need, and we're all thankful this gene therapy is available today. But the development program took place well in advance of the pandemic, before many of the current, digital health technology and other tools to decentralize could be done. So much of it had to be done in the clinic. As you would imagine, on the next slide, we wouldn't ask patients to come in to the clinic quite so frequently during a long-term follow-up, which is, of course, what we would be looking for any long-term follow-up to be achievable. And one quick example on the next slide, how would this apply to a theoretical gene therapy program for myasthenia gravis, which is a rare disease that is at the interface of immunology and neurology, where people can be diagnosed in childhood or adult. It's largely a chronic, non-lethal, but life-changing disease. We have, of course, another example where, because it's neurology, there are times when you need to have a manual expert evaluation for functional assessments, but we look to those well-validated patient-reported outcomes as well as digital tools and objective measures in any clinical trial that would be attractive to the MG patient community. And reinforcing the point on the next slide, we would, of course, ask less of those participants during the long-term follow-up, decentralized to an even greater degree if possible. So, how do you develop an end-to-end customized solution for your specific clinical trial? As we see on the next slide, the answer is you ask the experts, some of whom are the patients themselves and their advocates. Doing this most effectively benefits from specific expertise and on understanding the best practices in the space. Partnering with the community before your protocol is completely finalized is a powerful method for reducing risk. It's never too early to partner with the patient community, mapping out your clinical development program. They can provide insights at every stage of development. So what we see here is that clinical development life cycle from preclinical all the way through what we hope to be a post-marketing long-term follow-up phase. The subset of ways to partner with and the indication-specific patient community are sometimes obvious, and there are steps that have become standard practice, such as leveraging the natural history database and patient registries that many patient advocacy organizations have developed. But the final planning stages of an interventional phase, especially once a CRO has been engaged, are also an opportunity to collect and analyze qualitative feedback from patients and their advocates. Things absolutely do need to move quickly at this stage. If the sponsor team or CRO or both have already started developing a relationship with the community, collecting and analyzing that qualitative feedback can be accomplished in literally just a few weeks sometimes. But by keeping this order of operations in mind at the outset, and it applies to every phase of clinical development. I'm showing it here on the left, but it's just as true for the later phases. it's possible to tailor the study plan to the needs of the patients we're looking to serve with the development program, thereby making the study more achievable without altering the critical path of timelines. So making the study more achievable will obviously help drive recruitment and retention, and it's what we're supposed to mean with buzzwords like patient centricity. It can be a new way of working for some teams, which is understandably makes it feel complicated, but it's really fundamental project management of collecting the requirements at the beginning. But in this case, where we've recognized that making the study achievable really is a requirement, by developing these relationships early, it's an opportunity to not just support later recruitment and retention, but an opportunity to develop a reciprocal relationship where the patient advocacy organization can take a strategic role on input, on operations, and potentially even speaking on behalf of the study with regulators. Next slide, please. While I'm talking about starting as early as possible, that reciprocal relationship does support everybody, all parties throughout the development life cycle. When we involve the patient community in planning and design of clinical trials, they have higher confidence in how achievable participation can be and are a really important voice in the conversation with their own communities. So last winter, prior to the pandemic, one of our managers of patient advocacy and engagement within the Center for Rare Diseases, she and I got together and interviewed a number of patient advocacy organization leaders across the spectrum of therapeutic areas. And these were focused discussions on how we could support the success of gene therapy trials for all of their communities. And it was amazing to me how often these leaders highlighted the need for more and better patient-facing education about each gene therapy program as a tool for empowering families to make truly informed choices as they weigh that risk-benefit ratio. One that we have hopefully tilted in the right direction by taking that patient-centered approach to design and operations. So wrapping it all up on the next slide. Regulators really have proven themselves appropriately flexible and are broadcasting their commitment to continuing to develop regulatory framework for decentralized clinical trials. And every single gene therapy program will have unique needs, driven in part by the treatment population and the tools we use to measure that treatment effect. But in each case, there's a customized end-to-end solution for every gene therapy program, decentralized to the degree needed to meet the needs of all the stakeholders. We'll all benefit from asking the experts, the patients, their care circles, and community advocates. And that's it from me. I will now hand the microphone over to Chris.
Good day, everyone. Welcome to the second portion of our three-part webinar. Thank you, Amy, for an excellent first third, and looking forward to Christine after me here. My name is Chris. I'm a chairman of a regulatory services organization called Supply Global. We have a central IRB called CASTLE, Institutional Review Board, that reviews the ethics and informed consent, clinical biosafety services, which provides biosafety, institutional biosafety committee and GMO support, and last, a consulting arm for a variety of items to support gene therapy companies as they move from inception through clinical trials to FDA approved. Next slide, please. So for my portion, we're going to go through a baseline of where gene therapy is today in terms of investigational new drugs, some of the regulatory infrastructure and challenges with gene therapy trials, some of the institutional challenges, and because we act in a broad center for almost like Switzerland, in that we see many gene therapy trials across many sponsors, I want to provide a general clinical trial lessons learned examples. So, next slide, please. Before we get going, I do have a poll question. I'll turn it over to Ryan.
Yes, thank you very much. So as before, you can participate in this by selecting on any of the answers and then clicking submit. The question that we have for you this time is about how many gene therapy trials worldwide have been conducted or are ongoing. Your answer options are 250 to 500, 500 to 1,000, 1,000 to 2,000, or 2,000 to 4,000. We'll give you a few seconds to consider your answer and submit one. The question again is about how many gene therapy trials worldwide have been conducted or are ongoing. It looks like most of you have submitted an answer though, so thank you very much for your participation. Let's take a look at the results. Coming in for 38% at 500 to 1,000, 28% 250 to 500, 22% of you have selected 1,000 to 2,000, and right at the end, 12% for 2,000 to 4,000. Thanks again, and Chris, it's back to you.
Thank you, Ryan. Going on to the next slide. The source for our data here for the answer to our poll question is the American Society of Gene and Cell Therapy. They have a database that screens against the clinicaltrials.gov for the active number of trials. And on the mid-upper right, and a small number, you'll see here a 3231. So if you can go to the next slide, you'll see that our answer is D, or for the 2,000 to 4,000. The slide I'm showing here today shows INDs for gene and cell therapy trials since their inception back in 1989 at NIH. How there was a steady growth of about 40 to 50 new drug applications into the CBER division of the FDA from about 1990 through to about 2015. And it's really within the last 5 years that the methods for delivery, viral vectors, AAV, CAR T have really started taking off. And so we're looking at the last 2017, 2018, and we're waiting on data for 2019 and 2020. But there are over 800 active INDs currently being investigated under review through the FDA as of 2019, and we expect that number to continue to increase. Next slide. So the promise of gene therapy has always been, and if you can hit the next button here, that it's the future of medicine, that genetic engineering is going to change how we treat disease, specifically rare disease, oncology, vaccines. We've seen some of the technologies like mRNA used in some of the COVID vaccines, and that's been the promise. And so when you say gene therapy to a lay person or gene therapy to family or friends, there is an implicit knowledge that it's cutting edge, that it has the potential to do what the promise has always been, which is cure or drastically treat rare disease and oncology. So next slide. The reality of that effort has been, as you can see from the INDs, and go ahead and hit next on the next picture here, is that it's a lot of work, and it hasn't been as simple as we have thought. We have seen setbacks. We have seen that the challenges for biologics and their growth and development has been with many steps along the way, including some that I'll outline here on this webinar, but some of the many including the CMC, which is the chemistry, manufacturing, controls that look at how do we build out the biologics into an actual vial for delivery to the patient, along with simply getting these trials, getting them set up in a way that these patients, who many of them are under limited mobility to be able to get to these studies, how do we get these trials in as efficient a way to those individuals. Next slide, please. So one of the items that we did is we pulled together a few years back some of the leading luminaries in the gene and cell therapy space. One gentleman, Carl June at UPenn, is one of the fathers of CAR T and gene therapy. Another, Hans-Peter Kim, who's the incoming president for the American Society of Gene and Cell Therapy. And we published a article in Molecular Therapy of the American Society of Gene and Cell Therapy about what are the biggest challenges with these types of trials. And we pulled them together in a workshop, and we asked them what were some of the biggest issues they saw as a principal investigator. In addition, we also brought sponsors chief medical officers who run the sponsor side, and asked them what did they see as the biggest challenges as well, and highlighted seven pieces of the biggest challenges of these studies. Next slide, please. And in those, it was one of the big items outside of contracting is the length of time associated with regulatory reviews, whether they're national, local, IBC, biosafety, GMO for the specific countries, or the IRB, ethics board, REB for U.S. or international ethics boards. A lot of limited expertise and experience for these types of studies on these local IBCs and IRB panels. There's actually quite a bit of shortage in the biopharmaceutical space of folks who have experience with these types of challenges. So there's a growing number of these companies, but a limited number of experienced clinical operations team members who have done past work in this field, and so they're having to learn on the fly a whole net set of new regulatory requirements while also getting these trials off the ground. There's a public perception challenge with gene therapy. It is one of the more heavily scrutinized, both in the public sphere as well as regulatory sphere. The oversight of these trials in the U.S. is not always subject to a document called the NIH guidelines, and so there have also been regulatory changes to those NIH guidelines that have been confusing for sponsors and patients alike. Next slide, please. And so one of the other biggest challenges, in addition to the from the PI and sponsor side or principal investigator side for these studies, is also the challenge at each institution. Some institutions have quite a bit of experience with handling gene therapy products. They have SOPs, standard operating procedures, and trained investigational drug service pharmacists that are used to handling 10, 20, 30 drugs a day, a week in their IDS pharmacies. But as the products move from phase 1 into phase 2 and phase 3, more and more institutions were being pulled in, and fewer of these institutions had experience over the last few years and required a lot of education, training, and really the biggest one that we found was with pharmacies. And so collaboration between sponsors, Nationwide Children's, and Cleveland Clinic, we published an article in the Journal of Pharmacy Practice called the Gene Replacement Therapy, a Primer for the Health System Pharmacist, about what does it mean to be handling a biologic, a virus, a CAR T, a cellular therapy. What are the risks? What are the challenges? And go ahead and go to the next slide, Ryan. And what are the specific issues that these pharmacies have? And so the biggest item is because that gene therapy is classified still as a hazardous drug for pharmacies, that there's a lot of confusion on how they can prepare the drug, what do they need to do for cleaning and disinfecting the drug prior from working with chemotherapy compounds, so these biologics back to chemotherapy. There's a lot of lack of guidance from a lot of the organizations that are overseeing different aspects of how these biologics are administered, specifically to the pharmacy application. And then last, that while there is some information on the NIH of viruses and the risk groups, there's not a lot of information about what happens if a pregnant pharmacist is exposed to an AAV or a family member is exposed to a shedding from a recently dosed individual in case that they might need to have that treatment later in their life as well. And so these institutions have to come up with their own policies and procedures, which are very different and can often be very challenging to siphon through between the CRO, sponsor, as well as family members, because you might have access to 2 or more institutions that can dose and find that they have different setups for how they will actually dose you because each institution has different policies. Next slide, please. So a few clinical trial lessons that we've learned that I wanted to share as some examples. So again, I mentioned this as we sit as an organization at a crux where we see many different sponsors and gene therapy trials, and along the way we've seen a few items that continue to occur. So we wanted to highlight those. Next slide, please, Ryan, for this talk today. But number one, expect the unexpected. You can have all your plans 2 years out. You're going to have your first subject in. You're going to enroll the first patient in. But between the chemistry and manufacturing controls, between natural history studies, between development of the protocol, manufacturing, something will happen. And the challenge is, how do you pivot at that moment? Not from the initial plans that you set out when the company receives its Series A funding and initial IPO, but what happens when you have the first challenge or setback? And so that's one item I point out. Go to the next slide, Ryan, and we'll go through some of the others specifically. One of the biggest challenges I've seen is a lot of sponsors, especially in rare disease and ultra-rare disease, they are trying to open multiple sites in multiple countries simultaneously, usually 1 or 2 sites in the countries that they're going to. And where they get hung up is on the national level submissions that are constantly changing country to country. So we've learned that Canada has recently gone through some changes, Spain's gone through some changes in recent years, and other countries are just now getting to the point where they're actually enacting certain GMO or biologic rules for the federal or national level submissions. And then on the local point is that some countries have the equivalent of these biosafety committees. They have GMO committees for the specific handling of these products. And that's been a challenge for many sponsors, is how do you determine which countries have those requirements, which don't, and finding out that information in advance so that you can put together some sort of study startup timeline that makes sense so that you can start to plan and tell folks when your trial will be open for those community organizations like Christine's going to talk about. Patient population, some of the logistics challenges. Do you go to your patients and open up lots of sites, or do you open up a hub and spoke model and bring them to you, or do you have the decentralized model that Amy was discussing? That's been a huge challenge for many sponsors, is how many sites do you open, how do you go to, and also convincing the principal investigators that if they're not a dosing center, that they still provide a tremendous amount of value by being able to see the patients who have recently been dosed. Manufacturing. One of the biggest challenges right now is making enough drugs of these AAV products specifically for rare disease. So do you, as an organization, build out your manufacturing space, or do you outsource to the organizations that currently have a year or more backlog? And how do you adjust that and deal with the cost with that? Timing. How much has your organization promised that you will get subjects in by? Because that will also make a big difference on the type of sites you go to. Your planning is what is the speed at which that you're aiming for. And that's a challenging question. And then the last I wanted to, on this slide, these trials do not happen just by a sponsor and a CRO. They have to be, they're a huge team of vendors and support services ranging from an IRB, a biosafety committee, to medical imaging, medical monitoring, data analytics, data statistics. There's a tremendous number of partners, and if you don't choose the right partner, that can also slow down your trial in ways that can't be foreseen as well. So go to the next slide, please. The other aspect that's been interesting is facilitating the new regulatory pathways for biologics in U.S. and ex-U.S. So government agencies, especially the U.S. FDA, has actually done a really good job of being flexible on allowing for fast-track designations or getting meetings in place to discuss how your drug can get through to an approval quickly. So that's been a really positive sign to see the FDA really bending over backwards for that. Another aspect I mentioned already earlier is the training of pharmacists and sites to handle these products. So as you go more and more ex-US of many institutions, this will be their first or second time handling these products. There's a bit of concern for those staff handling these products, and it require some additional on-site or off-site training. And then remembering that accelerating the study startup is, as time is precious for these patients and their families. They're watching to see what your competitors are doing as a sponsor to see if their drug trial is coming through. They're talking to each other, and they should. They're parents who are trying to find the information to help treat their children. So they are not going to wait around for you, and you need to reach out to those community organizations, be very transparent on your timelines for them, especially for these rare and ultra-rare families who are working as hard as they can, and hear from one such example here in a few minutes on their to treat their children or treat their family members. And so you have to be cognizant that you have to be very transparent to those organizations. Next slide, please. So I just show this example. This is typically where a gene therapy product is prepared. You'll see that it's vented out through the ceiling. It's handling what's called a biological safety cabinet. That's a type of glove box. It's pharmacists wearing personal protective equipment, including a hair bonnet, mask, gloves, and operating through a glove system. And then they transfer the product out through the transfer pathway there, and then it's transferred up to either an OR neurosurgical suite for injection. But I just show that because these hoods, these units, many of them are 24/7 being handled for chemotherapy. So in order to clean these hoods for a biologic, it can shut down the pharmacy for a half hour to 45 minutes. And so we have to plan for that timing and let the IDS pharmacies know that they need to allocate that time. So I just thought a visual here would be helpful. Next slide, please. So in conclusion here, the gene therapy platforms, AAV and CRISPR especially, we're going to see a tremendous amount of increase in trials for rare disease and ultra-rare disease. They lower the cost by spreading R&D and making rare disease and ultra-rare disease treatments and potential cures possible. The end goal is it's worth it. This is for the patients and their families. It's going to be a lot of work. but at the same time, being able to treat subjects like my son or other individuals who we'll hear about in just a minute with these types of treatments, it's amazing where the science has come in such a short amount of time. The challenge is pulling all those pieces together as quickly as possible and as safely as possible. With that, I'll end my session and turn it over to my esteemed colleague, Christine.
Hi, everyone. My name is Christine Wagner, and I want to thank Amy Raymond and PRA Sciences for inviting me to share our story, and it's a privilege to share it today. So next slide, please. My poll question is going to be about GM1. So I'm just curious about how many people are familiar with it.
Yes, so as with the previous two, just like this one, you've got a question in front of you, and it asks, could you describe the rare disease GM1 gangliosidosis to anyone in your daily life? Your answer options are a yes or a no. We appreciate you taking the time to consider and provide an answer. It will help to set up our presentation as we continue today. It looks like most of you have submitted an answer. Thank you very much. Let's take a look at your results. Interestingly, 86% have come back saying no with a 14% at yes. So thanks again, and it's back to you, Christine.
Thank you. So GM1 gangliosidosis is a lysosomal storage disease, so a metabolic disease caused by the deficiency of beta-galactosidase. It has an incidence of 1 in 100 to 200,000, and it is a catastrophic neurodegenerative chronic and fatal disease. This is my sweet Iris. That is the name of her website, sweetiris.org. She's my daughter. She was diagnosed at age 5.5 in August of 2013. And we have spent a great deal of time setting up our nonprofit, engaging in patient advocacy. And so you can see the progression of the disease here in her and her story. So at age 10, she was using a walker. She is now age 12. She'll be age 13 in a couple of weeks, and she is in a wheelchair and has lost speech and many abilities. I am very fortunate to have a wonderful family. I founded the Cure GM1 Foundation, and my husband has been incredibly supportive, and we also have a wonderful board and many advisors. I'm very fortunate that my son does not have GM1 gangliosidosis. Next slide, please. So just to give you a little background about our story, I wanted to share a very special video and song about what it's like to be a rare disease patient. And this video and song was composed specifically for my daughter. And I think it's pretty special because there aren't very many songs about the rare disease experience, and it is performed by another GM1 community member and singer and songwriter. So please play the video.